I will be talking about transcriptional regulation or gene regulation in general. So my name is Chongzhi Zang. I'm associate professor from the University of Virginia and also affiliated with our cancer center. So I'm a computational biologist. My lab focuses on developing computational methods and use data science approaches to study chromatin biology and transcriptional regulation. So the chromatin and all the factors interacting with the genome to affect the gene expression, that's the key biological object that my lab is interested in. And we know over the past 2 decades or so, there are many high throughput technologies have been developed that generated tons amount of data. And my lab has, the research programs in my lab include 2 parts. So one part is that we develop methods, computational methods, to better analyze these high throughput data from different technologies with the hope to develop new tools and software tools for the community. And over the past few years, we developed several tools, and we love to give them nice names: BART, Selma, Patty from the Simpson family, and more recently Dory. So today's talk, I will be focusing on BART and its related tools. And the other arm of my lab's research focuses on using data-driven or data-inspired approaches to study biology, to integrate the data and use computational or data science approaches to look for new interesting biology from the data. So our recent interests include looking at from different aspects to look at single-cell data, as well as phase separation and transcriptional condensates. So just to give a little bit about a background. So we know, so this morning, Professor Wang gave a very nice introduction about RNA and gene expression and RNA-seq. So we know that gene expression or transcription is regulated by many factors in the chromatin, in the structure of the chromatin. And whether a gene is expressed or not in a given cell type is controlled or regulated by many factors. And one of the most important type of factors is called transcription factors. It's a type of protein that usually contains a DNA binding domain that can directly bind or interact with DNA, and they may recruit other proteins and other molecules to activate the transcription of a gene so that the gene can produce RNA transcript. And a transcription factor or other DNA-associated proteins, they will have a distributed along the very long genome. And using the sequencing-based technologies, we can measure the location of some of the particular transcription factor, a particular DNA-associated protein, their locations across the genome. And these information can be measured by technologies like ChIP-seq, which was developed back in 2007. And as we all know, RNA-seq has been one of the most widely used technology to study the level of transcription in certain cells. And RNA-seq was developed around 2008. And in a given cell systems, we're not necessarily always know that which transcription factors are active, are important in regulating RNA gene expression. And one way to tackle that problem is that when transcription factor bind to certain regions in the DNA, they usually either create or take advantage of the structure of a so-called accessible chromatin region, or a piece of DNA that are not wrapped by the nucleosome or histone structure, that are exposed to protein interactions. So these accessible chromatin regions or open chromatin regions can be measured using technologies like DNA-seq, developed back in 2008, and more commonly used technology called ATAC-seq, which was developed in 2013. So both technologies use enzymes that can go to the accessible chromatin and cleavage the DNA and get the DNA fragments, so that they can measure these either DNA-seq or tag-seq can measure where in the entire genome the DNA are open or chromatin are open, so that transcription factors can come and bind. As we all know that in recent years, single-cell technologies can provide multiple modalities of information that can help us study gene regulation in heterogeneous cell populations. And for single-cell technologies, we can measure multiple dimensions of information, such as surface protein. And as the if we look inside the cell, we can measure a tag-seq and a single-cell or single-nucleus RNA-seq in the single-cell matter. And there are other technologies such as single-cell multiome by the 10x Genomics company that can measure RNA and tag-seq profiles in each same cell. So using these technologies, we can also generate data and help us study gene regulation. So a common question that we want people are interested in when they study gene regulation is that how to predict transcription factors. So if we get the data and we have the data analyzed for single-cell data, for example, we can we can identify different clusters of cells that usually represents different cell types. And from these different clusters of cells, we can identify cell type specific expressed genes annotated by different highly expressed genes. And for both single-cell and bulk RNA-seq, one can identify differentially expressed genes using differential expression analysis. And once we got all of these, one common question we want to address is that, can we predict what potential transcription factors that are function to regulate a set of genes that are interested? So we have the expression data, we have a set of genes, and the question is what transcription factors may be active to regulate them. So how to identify functional transcription factors? There are many. So this is a question that's been there for decades. And there are many ways or different types of approaches to address this problem. And each type of approaches may have some limitations. So one commonly used approach is the gene ontology analysis, so which based on existing databases about factor and the target relations and the functional group relations of certain genes. And the idea is to find the enrichment of certain annotated gene sets in your gene set of interest. And of course, these ontology-based methods, such as gene set enrichment analysis, they are limited by the existing databases, limited by the existing knowledges. And another approach is co-expression-based approach. So by analyzing, by using so these type of approach has the assumption that the correlation between the regulator and their targets, the high correlation could indicate the regulatory relations. And the limitation of this co-expression based approaches is that in fact the expression of a transcription factor does not always necessarily mean the regulatory activity of this factor. And a transcription factor, even they're expressed, they may be regulating different targets in different contexts or in different biological conditions or states. And the third group of such methods is DNA sequence motif-based methods. Because over the years, when we study transcription factors, we may know their DNA binding domain, and we may have some idea about what DNA sequence patterns that each transcription factor is specifically bind to. So that once we have a gene set of interest, we can look at their, say, gene promoter regions and look at the relative enrichment of sequence motifs and to potentially predict potential transcription factors that bind to this motif to regulate gene expression. However, some limitations, such as the the motif occurrence does not always mean the actual transcription factor is bind there. And usually this approach is difficult to deal with distal enhancers. In mammalian cell systems like human or mouse cells, most transcriptional regulation or transcription factor binding actually happens at distal enhancers, relatively far from the gene promoter region. So then the direct look for the regulatory region may not be a direct, straightforward problem. So a few years ago, my lab developed a method we call them BART, standing for Binding Analysis for Regulation of Transcription. Transcription. So the idea is to take advantage of hundreds or thousands of publicly available sequencing-based data, including DNA-seq, which measures open chromatin, that we use this information to construct the regulatory region repertoire from the genome. And we also collected over 1,000 ChIP-seq data for a histone modification, H3 lysine 27 acetylation. So this is a histone modification that specifically mark active enhancer regions as compared to other histone modifications that also correlated with gene expression with different functional association. So we use the H3K27 acetylation ChIP-seq to quantify the enhancer activity. So the first step of the BART algorithm is that our input is a gene set, and the goal is to predict what transcription factors are regulating this set of genes. So by looking at the enhancer activities at these potential regulatory regions, and comparing by looking at such data from thousands of different data sets, we use adaptive lasso regression plus a semi-supervised learning approach to generate a cis-regulatory profile or a ranked list of all potential regulatory regions that are functioned to regulate the potential target gene sets. And then with these cis-regulatory profile, the next step is to compare this profile with the ChIP-seq data. We also collected over 13,000 ChIP-seq data for nearly 1,000 different transcription factors, and to identify those transcription factors whose genome-wide binding profile are most associated or most correlated with the predicted cis-regulatory profile. And we think that this way we can identify the transcription factors that are regulating the input gene sets. And then over the years, we have expanded and maintained this algorithm as a open source tools as well as a web server, so that they can take different types of user input, not only from a gene set, but also can directly from a tag-seq or ChIP-seq profile, or just a set of genes in the genome, and an extension of its method that can also analyze high-C or 3D genome high-C data. With all of those, with the goals to identify transcription factors that have the binding profile that are most associated with a regulatory profile of interest. Over the years, this tool, BART, has been used by many labs, experimentalist biologists, to study their interesting transcription regulation systems in their biological systems and make interesting discoveries. And these are just two examples of papers published in Nature that both use BART to identify interesting transcription factors in their respective systems. So we're always happy to see that others used our tool to make important discoveries to study biological systems. And a recent review paper or benchmark study published last year, they did a benchmark of transcription factor prediction tools. They include both just as DNA sequence motif-based tool, as well as other similar tools that also use collected ChIP-seq data. And I'm very excited to see that they rank BART as the top among all the tools. In fact, there are a few other similar tools that they all have good performance. So indicating that these tools can be indeed be used to make accurate predictions. So now the question is how to predict functional transcription factors from single-cell data. So initially, all of those is just an gene set input. Most of them are from bulk analysis. And although we did not develop a tool specific for single cell, we saw other researchers are already using BART to their single cell RNA-seq data sets, and they identify interesting transcription factors regulating different cell types. And this is an example this is a study published in Cell Reports a few years ago, in which they identified different subsets, different group of tumor-associated macrophages, and identified transcription factors that mark each of these subtypes of tumor-associated macrophages. So my graduate student, Hongpan Zhang, took the lead and to update the BART methods and developed a new tool called BART. We call this BART SC, or BART for single cell. It's an extension of the BART method specifically designed for single cell data. So the idea is still very similar to BART, but for single cell data, we hope this tool can be able to analyze either monomodal or bimodal data, such as single-cell RNA-seq, single-cell tag-seq, or and single-cell RNA and tag multiome data. So the idea is we can analyze or pre-process the single-cell data using any existing single-cell processing software to generate cell clusters. So after we get the clustered single-cell data, we also want to identify the features or gene sets that either be the cell type specific signature gene sets, or we can do a pairwise comparison of each pair of across each pair of the clusters to identify the differential features. And then for both features, we can just apply BART to identify potential transcriptional regulators. In this context, we call them the cell type signature analysis or cross-cell type analysis. So the cell type signature analysis is very straightforward. It's just each cluster, each cell type, they have a gene signature and apply BART to identify the TF specific for that cell type. But for cross-cell type analysis, Hongpan did a little bit further. So we first identify pairwise differential gene signatures by comparing each pair of gene clusters. And then for each transcription factor, we can identify their relative enrichment or relative activity comparing each pair of cell clusters. And then so now for each transcription factor, we can get a two-dimensional matrix measuring, with each element measuring the deviation. We call this deviation ratio, or we can consider that as the relative enrichment of the activity comparing cell Z versus cell X. And then for this transcription factor, we can give them a quantification about what this transcription factor's activity comparing to every other cell clusters. And using this approach, we give another matrix for each transcription factor. And we found that this transcription factor, in some cases, they can complement the approach of simply looking at the expression level of the transcription factor itself. So in some cases, although the transcription factor may not be specifically expressed in that cell type, but using this approach, we can find the cell type specific activity in a certain cell type. So then we integrate different matrix to give a final prediction about BART-SC single cell predicted, we call this cell cluster specific key regulators. So these three metrics include the signature contribution, so which means that directly use the cell from each signature genes from each cell clusters, and the relative activity by pairwise analysis, and also adding a third matrix about uniqueness to see whether this factor is unique for each compared to this enrichment in other cell clusters. And by integrating the three, we can give the final prediction of transcription factors. And we want to use some well-studied cell system to prove that our method works and have accurate prediction performance. So we use the PBMC, which basically contains a lot of known blood cells, including immune cells, B cell, T cells, and natural killer monocytes and dendritic cells, as well as some subtypes that some of them are shown in previous slides. And for each cell type, we manually curated these cell type specific key regulators. And we use these curated key regulators as the ground truths to benchmark our tool as well as several other tools to see whether they can make a correct prediction. So comparing BART-SC with several other methods that can do similar transcription factor analysis from single cell data, we found that among these 5 cell types in PBMC, overall, BART-SC has the best performance in terms of both F1 score and this sensitivity combines, if you look at sensitivity and specificity separately. So then we also tried this on another single cell data from cancer patients. So this is a single cell multi-omic data from a pancreatic cancer samples. And first, we show that we can see that BART-SC can identify transcription factors that's specific to each annotated cell type from this data. So by the way, we use their published annotation of different cell types as the ground truth for cell type, just for cell type annotation. And similarly, we manually curated the actual transcription factors that's specific to each of the annotated cell type. And we found that compared to other tools, BART-SC has these actual transcription factors always rank on top among relatively has the highest rank compared to other transcription factors. So now let's move on from a single cell to spatial omics. I love this diagram to show that the importance of spatial technology that can not only give us the information of each different cell types, but also their spatial information. So how to predict functional transcription factors from spatial omics map data? So this work is done by my rising second year PhD student, Jingyi Wang, who is also at the conference. So BART, we call this BART spatial, BART specifically for spatial data. And so the key question for BART spatial is that how to give a input feature or input gene sets that so the BART to be able to predict transcription factors. So we know that there are many computational tools that can identify spatially variable genes from spatial transcriptomic data. And if we treat the spatial transcriptomic data as single cell data, we can also perform cluster or trajectory analysis to identify genes that are also variable across the trajectory or the so-called pseudotime. And the idea is to integrate the genes from both analysis. So these are the gene features that show both pseudotime or temporal variable feature as well as spatial variant feature, and use these genes as input to identify transcription factors that are regulating these spatially variable genes. Because of time, we just give one case study, one example. So this is a spatial transcriptomic data from a mouse brain, developing mouse brain tissue. And we specifically look at, based on their published annotation, we specifically look at these regions that mark 2 cell types. And this is a cell differentiation states from radioglia to post-mitotic premature neurons. And so this is their published cell type annotation. And by using this spatial or trajectory analysis, we can label their relative pseudotime. And we can see that indeed, from one cell type, there's a differentiation potential to the other cell type. And this is just the spatial map of example of some spatially variable genes. And using these identified spatial variable genes as input, we're able to identify transcription factors that are active in both upstream or downstream, meaning they are active either in the radioglia or in the post-mitotic premature neurons. So for these highly predicted transcription factors, or some of them, we can see that the expression level of these predicted transcription factors also show a clear spatial pattern that are highly expressed in radioglia compared to premature neuron, or the other way, and as correlated with the pseudotime using RNA. However, we also found some other highly predicted transcription factors that do not show this pattern. So KLI4 is one example. So this is transcription factor predicted as highly as they are active in the upstream or in the radioglia. But if we see the spatial map, the expression level of KLI4 itself does not show the spatial variable pattern as other factors. Fortunately, we also found the matching spatial ataxic data from this publication, and we found that although the expression of KLI4 transcription factor itself do not show spatial variable activity, if we look at the differential or spatially variable open chromatin regions from the spatial ataxic data, we can find that KLI4 motif is enriched in the open chromatin regions that are specifically accessible in the radioglia. Also, then if we look at all the KLI4 motif containing open chromatin region, we can also show a differential level of the ataxic or open chromatin level in their spatial map. So this also indicates that although the gene expression of the expression level of transcription factor itself may not be directly informative to infer their target genes, but their activity in the chromatin is a more important information to give us transcriptional regulation patterns. So in summary, I hope this small lecture will convince you that identifying functional transcription factors is an important task to study gene regulation. And the key idea of these BART tools is to take advantage of public available data, which contain very rich information for functional functional and regulatory genomics. And based on the BART algorithms we developed before, I showed you two extensions or two other versions of the tools, BART SC and BART Spatial, which can we can work to predict active transcription factors from single cell data or spatial omics data, respectively. So with that, I would like to end here and thank our lab members, including the people who did the work, Hongpan, Jingyi, and others, and as well as collaborators and funding resources. I don't know, maybe we'll have some time to answer one or two questions. Thank you very much.